Hi, welcome to the lecture on inward adaptation of animals. We will continue now the series of circadian rhythm, and we already know the basic mechanisms how to keep circadian rhythm or daily rhythm in the cell. You need few proteins that are, for example, phosphorylating each other, and then they are activating or inactivating them. So, and there's this negative feedback. And usually there's also related that, okay, one of these proteins is affecting on the gene activity, and then there's a messenger RNA coded by this gene, and then there is a protein that is then inactivating the regulator. So we know that now the basic mechanisms, and now we are going on the location. And in the vertebrates, it's called suprachiasmatic nucleus. Suprachiasmatic nucleus is a pretty complicated word, but it's understandable that when we remember that, okay, it's located in the place where the neural tracks from the eyes are crossed. So behind your eyes, there goes white tracks where neurons are going to the central nervous system, and they will cross in the place called optic chiasma. And above this optic chiasma, there's suprachiasmatic nucleus, very small one, and it's then understandable that, okay, some information from the eyes goes easily to this place. And then if you are marking where these clock gene expressions happened, for example, where the per protein exists, they exist in this animal only or mainly in the brains in this place called suprachiasmatic nucleus. And then if you remove or destroy the suprachiasmatic nucleus, you will lose completely the daily rhythm. For example, in this golden hamster, when having a free running rhythm, so it's completely in darkness, and so it means that, okay, it tries to keep the daily rhythm with this with the clock, but because the clock is a little bit going too fast or a little bit too slow, then it can't keep the rhythm completely, and that's why there's a delay. So after the first day, on the second day, it's a little bit later, and the third day, it's a little bit later, etc. And then during the day 25, when the suprachiasmatic nucleus was destroyed, the activity is completely chaotic. So there's not anymore any kind of rhythm that, okay, now the animal is active and now it's resting. And it should be active on the second day at the same time. So that's why the suprachiasmatic nucleus has something to do with keeping our daily rhythm. But the suprachiasmatic nucleus is not the only clock. There are several clock cells that are receiving the light information from the eyes with the retinohypothalamic tract. So it's a little bit different tract than going in our cerebral cortex, but it coordinates the daily oscillation of other tissues. So it gives the information for brains and kidneys and liver, and all these other tissues, they also have some clocks, but these clocks are under the demand or under the order from the suprachiasmatic nucleus. And if you isolate a single neuron from suprachiasmatic nucleus, there is a robust circadian rhythm. But if you are isolating a tissue sample where there is a lot of these cells, so they are synchronizing each other, then you can see a measurable rhythm that can be maintained for weeks if you have removed the suprachiasmatic nucleus from the animal. So the suprachiasmatic nucleus is the predominant clock, but beside that, in several other tissues, there is this kind of fluctuation. and this fluctuation is happening in 24-hour rhythm or whatever. But in the periphery, these rhythms are usually lost after isolating the tissues. So the bioluminescence that is used for these kind of measurements in the ovary or the kidneys or pituitary gland and etc., there's a high rhythm, but it will be lost quite drastically within, for example, 1 week. So they are secondary clocks that are under the control of this main clock in the suprachiasmatic nucleus. And okay, that means then understandable that okay, that's why we have so many different physiological phenomena that are regulated by the daily rhythm. So we have higher pressure in the circulation at a certain time of the day. We are releasing urea at certain time of the day, and we have hormones are released at certain time of the day, etc. But which genes are regulated? And that's quite a lot. In this excellent study in 2018, they were having a quite large study relating on this. They were studying 25,000 different transcripts from the genome of a monkey and tested them in 64 different tissues and then at different time of the day. And then seeing that, okay, are there any cyclic genes other than these clock genes in suprachiasmatic nucleus? And there exist. In the suprachiasmatic nucleus, there's about 100 different genes that have this rhythm, and they are mostly active in the early in the morning or at the night. And then, for example, in the guts, in the ileum, they have 2 peaks, and in these there's about 400 different genes that are activated. Then in the heart there's over 300 different genes that are activated, etc. So we have in different tissues we have a huge amount of genes that are activated, and there's a lot of transcription. And this is a quite interesting way of make transcriptomics. So we have a cyclic expression, and interesting was that okay, of the 18,000 protein coding genes, 80% has daily cycle in the gene expression, at least at some tissues. And there are some circadian rhythm can be seen that okay, in this monkey at certain time of the day there is a plenty of gene activity at 1 o'clock in the morning, 7 o'clock in the morning, and then perhaps 11 o'clock at the night. And so there's a rhythm going on not only in the brains but in practically all kind of tissues. And interestingly, the amount of cyclic gene expression is not related on the total gene activity. So there can be tissues where there's several thousand of genes are active, but there's only few of these cyclic genes, also these with the circadian rhythm. Or there can be cells where there's only few only limited number of genes that are active, but all of them have this kind of daily rhythm. And usually these cyclic genes are highly expressed. And interestingly, there is not a single gene that has this rhythm in all tissues. So although we have learned that in the suprachiasmatic nucleus we have the main clock with the per and tim and clock, etc., but in some other tissues they're using different mechanisms. And when they've been testing, over here in the left hand side you can see that in some cells there exist several thousand cyclic genes. So genes that are expressed more at certain time of the day. But usually these kind of cells, there's not plenty. There are only few different tissues. And then there are a huge amount of tissues where there are only one or two periodic genes, like PER1, BMAL1, or CRT. So there's over 50 tissue where the CRT is the only gene that has this circadian rhythm. And then on the right hand side you have the clock, and you can see that these commonly related to circadian rhythm genes, they are expressed at a different time of the day. So for example, PER1 and PER2, they are expressed at the one time, and then these, for example, CRIB1 is expressed at another time, etc. And then they are regulating different processes. So that's why, for example, DNA repair or lysosomes are lysosome-related gene expression are happening at the certain time of the day, and then 12 hours later there is genes that are related on the ubiquinity. And so we have several cell processes have different kind of rhythm in our daily life, and also they are related on different ways on different places in the cell, either on transportation from endoplasmic reticulum to Golgi, or a little bit less on the how to move the stuff inside the Golgi, etc. And so it's pretty amazing amount of genes that are expressed at differently at different time of the day, and that makes a very reasonable that okay, why practically everything is regulated with our daily rhythm. But what about then? We already learned that in the cyanobacteria there was a compensation of this circadian rhythm on the temperature, but it's also important for the wildlife animals. So because the biochemical processes are slower, so there's need some compensation, so otherwise the clock should be very slow and late. And in this fish they have 4 different mechanisms. First of all, they have this entertainment by the temperature cycle. So we can have upregulation or downregulation of the gene expression, and then what happens next for the tissues that they are controlling, we can enhance that in cold, or we can regulate the expression level of the clock genes also. And then we can have temperature compensation, so make the clock slower in warm. Or we can have effect on the amplitude of this cyclic expression. So ectotherms must be able to remain this circadian rhythm in all their temperatures, and not only during the temperatures, but also during the hibernation. So when comparing rat and squirrel that is hibernating, they have completely different firing rate in the suprachiasmatic nucleus neurons, and usually in the mammals the firing rate is decreased drastically when the temperature is reduced. But in the squirrel that is hibernating, it must be able to maintain the suprachiasmatic nucleus. And how it can be done, it's just because it has lower activation energy for this firing. So that means that, okay, then the Q10 is smaller in squirrel than in the rat, and that's why it can maintain it even below 20 degrees of Celsius. And that's the only way how this ground squirrel can work with the suprachiasmatic nucleus during the hibernation. Because now the body temperature is 5 degrees Celsius. So it would, if it would have the same kind of clock as the rat, it would never wake up because the suprachiasmatic nucleus would not work. And the hibernation has been studied actually quite a lot during the related on this circadian timing. So in the golden mantle ground squirrel, the turbots, the length of the daily hibernation are a little bit shorter in the beginning and end of the hibernation. And if you remove the suprachiasmatic nucleus, they will have a continuous torpor for 20 months. So never-ending hibernation. Of course, that's lethal. But it's also affecting on the dynamics. So how long they will last, these torpors are lasting, and also it's affecting on the intervals where they are aerothermic. And if you have a partial lesion, it's only affecting on one thing. So it seems to be that the suprachiasmatic nucleus and the daily rhythm is now explaining the hibernation that we had previously on this course. And it's also affecting on this circadian clock. So in most of these hibernating animals, they have a clear cyclic expression of these circadian genes, like PER1 and PER2. They are highly expressed when there is a sunlight. But actually it's a little bit more confusing. There's a study in European hamster that it will shut down the circadian clock during the hibernation, and no one knows how it can maintain or know that, okay, when to wake up from this hibernation. And it's also been speculated that this torpor can affect on the timing of the expression of these clock genes. But there are still things to do on this subject. Thank you.